Okay, today we have the award ceremony for the Evelyn Hutchinson Award, and this is to recognize excellence in any aspect of limnology or oceanography. James Elser is recognized this year with this award. He was nominated by James Collins, letters from Steve Carpenter, Susan Killam, and Dag Hessen. Brief citation, excerpt from the citation for Jim. James Elser is an outstanding scientist whose contributions to research, education, and service fit the ideals of the G. Evelyn Hutchinson Award. Jim couples innovation and a passion for discovery to make, as Hutchinson asked of his recipients of this award, considerable contributions to knowledge that ensure a legacy of scientific excellence. Professor Elser is an innovative, broad thinker whose ideas are changing biology by adapting, modifying, and creating theory to explain a broad range of phenomena. Ecological stoichiometry has the potential to be among a handful of unifying theories in biology as a result of the rigor and imagination Jim brings to his research. And I totally recommend that book. It was really, in my mind, very transformative in terms of taking the general Redfield approach to this kind of stoichiometry and incorporating it much more into interesting ecological and evolutionary ideas about why these elements change in organisms. James Elser deserves this year's G. Evelyn Hutchinson Award for work already completed, promise of work to come, and the living legacy that Jim is building through sustained and generous investment in his research, colleagues, and students. And I think this is a very, very special award. I, as a postdoc, had the opportunity to meet Hutchinson in his old age at the time, and I can't think of a better person more deserving of that. So Jim, please come up and let's congratulate Jim.
Thank you, Tom.
It's fine. You just need it.
That's fine. Well, thank you so much for coming. I have the remote microphone. It's going to be hard for me to stay in one place. I have to move around. So it's just a wonderful, wonderful thing to be here today. And the first thing I have to say, of course, is arigato gozaimasu. Thank you very much. Thank you, Tom. Thank you to the committee. Thank you to ASLO. Thank you to all of you for coming today. And so I have a few thoughts to share with you about what we've been doing and some other ideas about what needs to be done more broadly at the end. First, I want to explain my title to you. The title of my talk is Keep Walking to Kyoto and Back and Beyond. I didn't know what to say when you receive an award like this. I was just at a loss about what was appropriate to say. So I thought I would do my normal plan, which is to say something inappropriate. But instead, I decided against that, and I went to Hutchinson for inspiration. And you may know that Hutchinson received the Kyoto Prize in 1986, the highest award that outside of the Nobel Prize categories that can be won by a scientist. And this is him accepting this award in Kyoto in 1986. And the title of his acceptance address is called Keep Walking. And so I think it's just so very appropriate that I can receive the Hutchinson Award here in Kyoto. So I came to Kyoto to receive the award, but actually I'm walking back because I've been here before many times, and for the longest time was in 1998 when I came on the first part of my sabbatical leave with my family, and here we are in Lake Biwa, and here we are. Here's Jotaro Urabe, who you've seen on the stage today, and throughout the weekend, Michio Kumagai, who also is the co-organizer of this meeting. And you can see that they haven't changed at all since that time, but you can notice that I have changed a little bit. So it's a tremendous honor for me to walk back to Kyoto to come to this meeting, to help organize it and to receive this award as well. So now I'm going to talk about why we're walking, what our issues are as we walk through our scientific careers. And here's one thing you might think about. So here we are down here, the great community of scientists that are out there hoping to make real contributions. And it's likely to be the case that there's some limited number of good ideas and insights that are out there available about how the world works. That's because the world is only one way, right? There's only one way it's working, and you hope to find those ways that it works in your research. The problem we have, so here's the limited pool of good ideas and insights that are out there. The problem we have is this, is that Darwin got there first. And if almost any idea, a good idea you think you might have, probably Darwin already thought about that problem and solved it. And so you guys ought to read everything he wrote and before you think you have a new idea. Now, some of them did get through, though. The bad news of that is that Hutchinson and Redfield probably got most of the ones that are left that you might be interested in working on. So there's our problem. All right, so again, if you read Hutchinson, if you read Redfield, the brilliance of their reasoning is manifest, and it's hard to imagine that you could come up with something new and different that could possibly be valuable. So here we are waiting for a drop of insight to come down to us that hasn't already occurred to these brilliant giants of the past. So what to do? So here I am down there. So I decided, I think what we've been doing in stoichiometric work is to try to solve that problem is by mashing them up, sort of the combination of all of them. So maybe we can make progress by trying to integrate all of this insight that these brilliant scientists have brought together in the past and make some progress. And I think that's a little bit of the flavor of what we've been working on over these years. So here is the book that Tom mentioned earlier, Ecological Stoichiometry, with my co-author Bob Sterner. And this book is our initial attempt to sort of bring all these ideas together in a coherent way, and to see what they mean, and to make some predictions about the future. So by mashing up these ideas of biogeochemistry and ecosystems evolution, ecological interactions, and the insights that we have already into a single framework, and see if that is a worthwhile thing. So here's how it started. It's a story that starts a while ago. In 1985. This is the glaciers were further advanced at that time. It was a long, long time ago, and this is work I did in one of the first experiments testing the theory of cascading trophic interactions of Steve Carpenter and Jim Kitchell, and Monica Elser was also involved in this study. And interestingly, I'm proud to say that this paper won the 1988 ASLO 1990 Lindemann Awards, published in 1988. And what we found was something quite mysterious. In the course of doing a trophic cascade experiment where we manipulated the relative abundance of piscivore or planktivorous fishes in lakes in order to change the zooplankton community in order to see if it affected the algal productivity and biomass, we discovered that when you do that, you also change the relative availability of nitrogen and phosphorus that are available in the ecosystem. So in lakes with in the piscivore-dominated conditions of this Peter Lake, the phytoplankton are phosphorus limited. Over here, whether you ask them with enrichment experiments or use physiological data to show this. But in a lake dominated by planktivores, therefore that has a lot of copepods in its zooplankton community, you have phytoplankton that are limited by nitrogen instead of by phosphorus. And what we discovered is that when you change the zooplankton community structure by manipulating the food web, you move the ecosystem nutrient limitation in different directions. So despite the fact that we had no idea what was causing this really, we were able to publish it in LNO, and it was given this award, and it was very pleasing. Fortunately, at the same time, some other scientists of around the same age class were working and starting to think about very similar phenomena. And so in 1991, a paper was published by Norwegian colleagues, Dag Hessen and Tom Anderson's group, and they showed for the first time, I believe, that the major genera of freshwater crustacean zooplankton that we have in lakes differ in their body elemental composition in major ways. Copepods are nitrogen-rich, as opposed to Chloropsera and Daphnia are lower, relatively low in nitrogen, and especially we see major differences in phosphorus. Copepods are low in phosphorus, Daphnia is rich in phosphorus. So this seemed to make an interesting implication for understanding what we just saw. At the same time, Bob Sterner was starting to think about these things. He wrote a very elegant model of consumer-driven nutrient recycling in which it's possible to calculate the entropy ratio of recycled nutrients based on what they're eating and the stoichiometry, the entropy ratio of the consumer under different assumptions about assimilation efficiency. And what you can see is that for a given food entropy ratio, copepods recycle nutrients at a low entropy ratio, therefore they ought to make things sort of N-limited. Whereas Daphnia, even the very same food will recycle nutrients at a high entropy ratio. They should make things more phosphorus limited. And so this was a really cool paper when it came out, and this is 1990. It's all the same few years when all this is starting to happen. Now, fortunately, around this time, email was invented, and the students are saying, what? Yes, back then we mailed papers around, and papers were paper, and you mailed them around, and that's how you did things. And so Bob and I had already met, but we contacted Dag Hessen, who we had never met, by email, and began to talk to each other about what we were seeing, and we were able to bring a synthetic synthesis of our observations by taking our data from these Wisconsin lakes, Bob's nutrient recycling theory, and the data that Dag Hessen had produced about the stoichiometry of Daphnia and copepods to explain why it is that when you change the food web structure in a lake such that copepods shift we have copepods shifting to Daphnia, you can shift the phytoplankton from N limitation to P limitation and back in the other direction. So this was really very satisfying, and we were quite excited to be able to achieve this insight. But of course, we really shouldn't be entirely satisfied. We're not really finished yet. We're finished if we're just, biogeochemists or something like that, which is a great thing to be, of course, but it's really not the end of the story because, for example, you might then ask the question, why do different zooplankton have different entropy ratios? And might it have something to do with life history evolution, for example? So we can be inspired by the kind of questions that Darwin and Hutchinson would have asked if they were in this situation. So this led us to produce what we call the growth rate hypothesis, where we propose that the variations in elemental composition that we just talked about, which drive these sort of ecosystem-level biogeochemical impacts, are driven by underlying subcellular allocation shifts because ribosomal RNA is very phosphorus-rich, contains about 10% phosphorus by mass, and therefore shifts in the relative allocation of these major biochemical pools in response to growth rate, because to grow fast you need to allocate a lot to ribosome biogenesis. Evolutionary changes that impinge on growth rate are going to affect the stoichiometry of organisms and therefore their ecosystem impacts and functions in the way diagrammed in this figure. And we also, in a more modified version of our hypothesis, have ideas about how the variations in the structure of the ribosomal RNA genome are associated with these variations as well. I don't have much time to show you very much data. Here's a nice plot, though, that sort of summarizes a lot of how it comes out. Here's a bunch of data for the percentage of dry mass of an organism on the y-axis and the percentage of the dry mass of an organism that's contributed by phosphorus contained in RNA. And if all of the organism's phosphorus is in its RNA, you'll be on the 1-to-1 line, but you don't expect to be there because there's other places in the cell where you have phosphorus besides RNA. We expect to be over here, but we see something very nice, which is within the individual experiments, the groups that are shown here, we see intraspecific correlations and across, and each of those is a growth rate associated. These are fast-growing E. coli that are phosphorus and RNA rich. Then as you move in this direction, they're in chemostats and slower growth rates. They have less RNA, less phosphorus. And here's something quite beautiful that you never expect to see when you're an ecologist, and that is a high R-squared value. But there we are. If you fit data to this entire data set, the R-squared is nearly 90%, and the slope of that line is 1, which means we don't just have a correlation, we have an explanatory explanation in our hands. So that's that was a very fun and important result, I think. So that was our digging. So we dug down from the ecosystem level, and we kept going down, and we end up at the level of biochemicals, like this is a ribosome, and at the level of elements. Now, I don't think we need to keep digging. The Higgs boson may have something to do with all of this. I'm not going to go there. It's time to stop digging and move out and upward. And so we've been doing that. So that was sort of a historical perspective. I just want to say a few things about what we're doing now as we move outward, having dug down and come back up in different worlds. And so one thing we're working on is applying the growth rate hypothesis in the context of stoichiometry and the growth rate hypothesis in the context of cancer, because cancer cells grow fast and may in fact have higher phosphorus requirements and therefore may also be phosphorus limited. So we've been working a little bit on that. We've developed approaches that we call stoichiogenomics that take advantage of emerging genomic databases to ask questions about the elemental composition of the macromolecules of life, the protein structures, nitrogen use in proteins, sulfur use in proteins, and phosphorus and nitrogen use in DNA and RNA. And so that's a direction we've been working. Most of my work right now is going on in the context of microbial evolution and using stoichiometry to help us understand, for example, how phosphorus drives microbial evolution, speciation, and adaptation. And we're also working on applying stoichiometry in outer space in the context of the NASA astrobiology program. And the final thing that I've been working on quite a lot in the last several years is the issue of phosphorus sustainability, not just from the context of too much phosphorus going into lakes, but the idea that phosphorus for fertilizer production is relying on a finite mineral deposits. And so the question there becomes, how long will the finite supplies of phosphate rock be able to produce cheap fertilizers? All right, so this is, I think, what we've been working on, walking a safe way to walk from here to there, from this type of complexity to that one, and back. Now, this is a good thing because I think this is where biology needs to go. Now, if you were at the ASLO meeting in Santa Fe that Debbie and I organized, you may remember that Craig Venter gave the opening plenary talk, at my invitation, actually. And the first thing he said when he gave his opening plenary talk was that he had never heard of ASLO before I invited him to give a plenary talk. And I had to restrain Debbie from running up on stage and just slapping him. And I was a little bit peeved myself. But actually, after he said that, I said, this is great. This is fantastic. These guys don't know what's going on over here. So we can help them ask interesting and important questions, right? There's still a chance for us as ecosystem ecologists and ecologists in general to use these powerful tools, right, to understand these systems and use thinking that comes from over here to help in guiding research over here. Now, I hope I have time to speak about one more thing, and that is this topic that is inspired also by G. Evelyn Hutchinson. And in his address, he talked about knowing what one is talking about. And that speaks to the issue of why we're walking in the first place. Now, I'm not going to read this to you here except to say that what he says is that for a society to function properly, it's important, a primary requirement of achieving things we want to do as a society is that we should know what we are talking about. That is, we should have an awareness of what is happening around us. And so that speaks to a really classical post-Enlightenment sort of view of the role of science in society, in which you can imagine that citizenry is sort of have weakly held opinions and notions about what's going on around them, but their understanding of the state of nature, like how their body works or how the environment works, influence the decisions they make in their daily lives, and also influence the decisions that collective action makes in the context of democratically elected governments, for example. And the role of science, then, is that we hope that data and discoveries and insights influence the understanding that people have about what is going on, right? And therefore, their elected government acts in a certain way that's informed by real knowledge about how the body works and what's going on, for example, in the environment, and, for example, might even be interested in asking scientists for input about what to do in light of that understanding. For example, Benjamin Franklin, one of the founding fathers of the United States of America, created the National Academies of Science to advise the government about the current status of scientific issues and to make recommendations for action. Now, that's a beautiful thing, and that's how I sort of saw science and society and thought that's kind of how it works. And it may often work like that, but actually it may not work that way. So here's a parade of the heroes of science, and I'm going to change it a little bit here, because here is actually the people who really make determinations about what's going on in the world. And I tried to be fair to the left and the right and put ideologues of all kinds on this little graph, because what's really happening is something that's called motivated reasoning, where people selectively seek evidence to support already formed opinion. And an example of this is the controversy over President Obama's origin of birth. And so motivated reasoning now being studied by cognitive psychologists as the way that people actually come to understand the world around me. And it looks more like this. They don't have loosely held opinions early on. They have opinions that they've grasped onto, and very often what happens is that they behave in ways that serve to strengthen those opinions. They build barriers to information flow. They will just pay attention to the data and the insights that agree with their ideological position. They will, of course, not really be interested in gaining advice of scientists because they already know what they're going to do, because it's based on their ideological position. So this is, I think, perhaps a more accurate view of what is going on in many places. I think this is going on in the United States now. For example, the state of South Carolina just passed a law saying that coastal planners cannot use the most modern projections of coastal sea level rise that predict an exponential increase in sea level in their planning. They are legally required to use only a linear extrapolation of the past. So that's an action where these actions that society takes place is to break off the data, the very production of information that could lead to disagreements of their preconceived opinions, and to turn off the role of government in all of this. So how do we keep walking in the face of that? How do we keep going? How do we make some sense out of that? And I don't know. Maybe I'll know in 30 years, but I think we need to completely rethink how we talk about science, how we connect to students, how we connect to the public. It's not enough just to explain it clearly, right? If explaining it clearly was enough to make that people will accept it as a valid view of nature, right, then more than 30% of the population of the United States would accept the scientific view of evolution. Only 30% of the people, or less, of the people in the United States accept scientific evolution. In Japan, 95% of the people accept the scientific view of evolution. So obviously there's something going on besides how clearly evolutionary biologists have explained the problem, right? There's something else going on. So I just want to move on to the most important part of my talk, and that is to thank the people who've been involved. I have to thank Bob Sterner. This is, of course, here we are. We seem to share part of our hemisphere. It's really quite been a remarkable thing to work with him as a partner in all of this work over time. And so I have to thank Bob from the bottom of my heart for all the partnership that we've had over the years. And in writing that book, during my first semester of my first sabbatical was writing the book in Minnesota with Bob. The second semester of my first sabbatical year was spent here in Kyoto with Jotaro Urabe, and so I thank Jotaro for all his help over the years and contributions. My second sabbatical was spent in Oslo, Norway with Dag Hessen, and so I thank Dag tremendously for our interactions over time. I've also interacted tremendously with Tom Henderson in projects over the years, and a very important person that I should thank is Marcia Kyle, who was my technician for 12 years and produced many of the beautiful data. If there's any of my data are beautiful, it's because that she had her hands in them. And I do want to just say that, sometimes things happen in science and there's a lot of competition and people sort of fight together. The team here of Bob, Jotaro Dog and Tom here. Just working with these folks has been really an amazing thing. I have to thank the ASLO Lab. I've had some terrific students, undergraduates, graduate students, and postdocs over the years. Just a wonderful group. So I thank everyone that. I've collaborated with a lot of people around the world, and so I have to acknowledge the value and tremendous contributions of that. It's been a tremendous thing. I thank ASLO, which has nurtured my career since 1982 when I went to my first ASLO meeting. So I thank ASLO for everything they've done for me for all these years. I've had a series of tremendous mentors who've cared for me and taken care of me over the years. Steve Carpenter, who I studied with as an undergraduate when he was at the University of Notre Dame, and then worked with him later on the Trophic Cascade Project. My PhD advisor, Charles Goldman, is inspirational for me in getting started and really going further in limnology. And at Arizona State University, most of the time I've been there, my department chair has been Jim Collins, who's now the president of AIBS and a tremendous person. I come from a big family. That has an effect on you. I have 5 siblings, so I thank them for all the good they've done for me. My mother died of colon cancer some years ago, and that was one inspiration from the work we've been doing in the cancer. My father's a tremendous person and a great inspiration as well. We've got a couple of kids. These guys have carried a lot of water around lakes for me, and so I thank them for that, and they're both tremendous young men. And the most important person I need to thank today is my wife Monica, who's here today. We're coming up on our 30th anniversary, and she is a scientist. Well, she did a very good job over here in taking care of these wonderful kids and doing the bulk of that. She's a scientist, as you saw. She's an educator and trying to help with those issues I pointed to earlier, so I thank her for that. And more than that, she's a wonderful person and my inspiration. So thank you very much.